Hi, I hope this is not going to be weird, but what I'm trying to do, I'm trying to re-record a video that I made earlier, in that video I didn't do the right setting, so that means I've got my face but I don't have the slides, so I'm trying to put in the slides with the face, and I hope that works. So bear with me as I try and get this thing set up correctly. Alright? Where is it? Hello. Hi. So we're going into translation now. Look at this picture. Can you guess whether this is a eukaryote system or a prokaryote system? Can you? Well, if you look at this picture, you can see this is a DNA, right? This is the DNA. And get the cursor. Okay, that's the DNA. And that's the mRNA. That's the mRNA, that one. And these dots here are the ribosomes. So this is an example where mRNA, while it is being transcribed, translation is already taking place. So in a piece of DNA, there's multiple RNA being. It's not just one mRNA being produced there. It's just this. There's a continuous process, mRNA being produced, and translation is also a continuous process, right? So this picture this has come from a book called Principles of Genetics, Snusted and Simmons, right? So okay. So this slide just talks about the different types of amino acids. You'll see that it has a general structure. You have the amine group, you have the carboxyl group, and then this is what you call as the R side chain, and this R side chain determines what type of amino acid there is. You have the hydrophobic, you have the hydrophilic, you have the acidic, you have the basic, right? Again, this one talks about the ribosome. The structure of ribosome, the small subunit is made up of 16S RNA. You will also be reminded later that this is the part that has the Shine-Dalgano sequence. This is an RNA. This is the part that will bind to the mRNA, right? Through the Shine-Dalgano sequence. So basically, this is rRNA, mRNA is like a base pairing between these two RNA molecules. The larger ribosomes are made up of the 5S and 23S. Now again, 50S plus 30S, you get 70S. This is not weight, this is not mass, this is sedimentation coefficient, Svedberg S. So this is a 70S. And in eukaryotes, as expected, it gets more complicated. Okay. This slide just talks to you about how do you get rRNA. rRNA gene, well, they are transcribed as a single RNA, right? So you can see here, this is a single RNA, and then it is cleaved by endoribonucleases. Endo means inside, ribonuclease, but nuclease cuts DNA, so ribonuclease cuts the RNA type of nucleic acid. So here you cut different sections. This is a 16S RNA section. This is a tRNA molecule. This is 23S. 5. Why there's a tRNA is just the organization of the gene as such. A tRNA molecule, this is a 2D flattened structure. This is a 3D structure. This is where the area of where the amino acid is bound to. This is the anticodon. This is the one that pairs with the codon. And a unique feature of tRNA molecule is that it has modified bases. You can see somewhere around here. There you go. You have this side. This is a pseudouridine. You have pseudouridine. This is methylinosine. This is inosine. This is dimethylguanine. Now this comes from the normal, the usual nucleotides, and they have been modified. So that's why they're called modified bases. And usually it only occurs in tRNA molecules. An important part of tRNA molecule, if you see the next slide, in the next few slides, where is it? Okay, this slide. This one talks about the fact that when in the process of translation, the mRNA sequence will determine amino acid. But that is only determined that is only the term. I've got a meeting in a bit, but never mind. The codon will match the anticodon in the tRNA, then tRNA will have the amino acid. What this experiment says is that the system, the translation system does not check for the amino acid. That means to say, if this tRNA, in this case, it codes for cysteine, so the tRNA brings cysteine, but if you can chemically change the cysteine into alanine, right? This mRNA codon anticodon is still correct, and because of that, the system thinks that it is bringing the right mRNA. They're bringing the right amino acid. So that's to say that the translation mechanism only depends on mRNA and the codon anticodon interaction. It doesn't really look at the amino acid. So that means so there is a crucial, very important for each tRNA to be matched with the correct amino acid, and this is achieved by this enzyme called amino acyl tRNA synthetase. Enzymes are very specific. Enzymes normally just do one thing. They can, unless they have different compartments. In this case, this amino acyl tRNA synthetase, what it does is it matches the tRNA with an amino acid. So that means to say that you would expect to have at least 20 different amino acyl tRNA synthetases, one for each amino acid, so that it can match with the right tRNA molecule. Okay. This is where it says translation relies on complementary mRNA tRNA, codon anticodon. Ribosome doesn't look at the amino acid, and this is the experiment to prove it. If you hear that sound in the background, there's just staff doing some vacuum cleaning the office. Sorry about that. Okay, then we go on to look at the process of okay, stop now. The process of translation. I think this is quite self-explanatory. What you need to remember is the very general process. You have the ribosome binding. This is the Shine-Dalgarno sequence. You have the IFs, which are the initiation factors, and then you have the recruitment of the start tRNA that brings the FMAT and the anticodon that matches the AUG, and then once the large subunit joins the whole complex, now this is called the initiation complex, and this marks the beginning of translation. And this is the Shine-Dalgarno sequence pairing between the 16S RNA and the mRNA sequence. Then once you have that, it is quite automatic that the tRNA is placed in the P peptidyl acyl. This is A amino acyl site. So the next tRNA will go in, and then this tRNA is the one that matches the second codon, in this case a GCC. So what's important is IGC. Now the other function of the modified bases is that some of them can pair to more than one nucleotide. So here you'll see that you have I, inosine. Inosine can pair with C. If I'm not mistaken, inosine can also pair with G. I can't remember offhand. But yeah, so these modified bases have the ability to bind maybe not just one, but two different bases. The specificity, it's not like the A's and T's, G's and C's. That's very fixed. But in tRNA, in the process of translation, this occurs. So this is the whole process of. Then you have the EFs, which are the elongation factors. And then it continues. I don't think I need to explain that. And then you reach the end, and you have the RF. RF is the release factors. These are the ones that recognizes the stop codons, because stop codons do not have any anticodons. So these are released by release factors. Release factors bind in, and then it gives a signal to the ribosome that marks the end of translation, and everything will start to dissociate. So now you have a peptide forming. So then everything dissociates, and that marks the end of translation, and also that marks the end of this video. I'll see you in the next video. Bye bye.